And then it's going to be 11 through 13 because that kind of groups all of the transport stuff together, how we're transporting things. Okay? Next week's exercise, so we're in week 12, and let me go in chronological order. We're in week 12. We're going to actually start, right? This is 12? Yeah. We're actually in mitochondria that we're going to talk about today. And then on Friday, what I would like to do is actually give you guys a full work day on Friday for bioinformatics. Okay, so you can come in here and do it. I will be here anyways, or you can decide to meet with your group somewhere else if you want to. It's totally up to you. All right, but I'm going to give you that freedom, but I am going to warn you, sometimes people are like, oh, we're going to do something else, and then they're the only one that shows up. And that's a really frustrating thing. So just make sure that you are knowing where you're going and that, okay? And not working on it by yourself. Unless you want to. That's also an option. It's totally an option to work by yourself. Okay, protein localization in class is posted now, but please look for the review sheet to change because even after I posted it, I started changing the conditions. So it's called protein localization, but I've also included moving RNA out of the nucleus too. So this exercise right here, if you open it and read the description, it's a little bit different from the other ones where we built PowerPoints. This one is actually like get into a group and do a hand drawing of the process, okay? And you can do one hand drawing together and then everybody looks at it, you know, if you want to. But then you're also going to have like a short table that you submit that answers are, it's like the review sheet. So the review sheet lists off the molecular players of that transport process and then the role of each of those things. So just a short descriptor to define, you know, what the what is the signal recognition particle, stuff like that. Okay. So that's how timeline. We're going to talk about mitochondria today. We're going to take Friday as a work day on bioinformatics in here or off. If I'm in here with one group, I'm okay with that on Friday. Monday we'll get back together. That's the 17th. So we will either, I've not decided yet, we'll either talk about chloroplasts then and get it done and over with. Probably that. And then by Wednesday, we will have some work time for protein localization, and maybe by Friday we can present. Okay? All right. So just keeping those things. We're starting. I want to go over the timeline because I'm releasing some things to you guys to do, and I just want to make sure that you know where when we're doing that stuff. All right. If there are any questions, please let me know. Let's see. Any questions you can think of? Feeling okay-ish? Okay. So we finished moving proteins in the cell to like the endoplasmic reticulum and the nucleus. So now what we're going to talk about is moving proteins into the mitochondria, chloroplasts, and other stacked membrane things, and which is what a plastid is, and then peroxisomes. So that's what this group of slides is going to be. I'm going to cut off with mitochondria today, which this picture is actually a network of mitochondria within the cell. You don't realize they look like this little discrete little bean thing in the cell, right? But they are actually networked with each other. They kind of work together. Many different ones in the cell. Oh, I'm not plugged in. So before looking at how things are transported, we want to continue to talk about, we want to review what the mitochondria is doing. We've got a pretty good understanding of it, and we've had our little talks about it beforehand, but now what we're trying, we really have to think about where things are happening for how we're transporting the proteins, you know, where we're putting them, okay, because so we need to think about that. Remember that the metabolism is the powerhouse of the cell, So it is where we have the most metabolic energy produced. So this is where we're producing ATP. And we remember that process for that. So what are we doing? We are generating energy from both the breakdown of lipids. We never talk enough about that, but lipids are broken down and they can be fed into cellular respiration as well, and also the carbs, so carb catabolism, right? We're going to eventually talk about chloroplasts, and so just a reminder of what they do and what their role is in metabolism. This is where we have sunlight energy that generates adenosine triphosphate as well. And within the chloroplast, we also use oxidation reduction to build carbohydrates or sugar from CO2, so bringing that in. So really major sets of chemical reactions that are happening within these organelles, and so there's going to be some proteins that have to be transported into the insides of them, into their membranes, all of that stuff. So the mitochondria has two membranes, which makes it even more complicated. We have the outer membrane and the inner membrane, and then remember there's a space in between. So now that you've already got the picture of how we move things into the endoplasmic reticulum, you can see how this is going to be a little bit different, right? So we've got an outer membrane to get through. There's an intermembrane space, and then we've got an inner membrane to get into. The other thing about it is the inner membrane is really there's a lot of it. It's folded. We have the cristae, right? So there are these other pockets of intermembrane space as well within there. I like this picture because, just thinking about how the mitochondria kind of work within the cell, keep in mind that there are several within the cell. All right, back into the mitochondria itself, we have the matrix that's inside. There's a lot going on. So the matrix is going to have its own genetic system and the enzymes for oxidative metabolism. So all of that oxidative phosphorylation, that whole part of cellular respiration, that's happening right in this membrane. And there are enzymes inside this space that are involved with that. And then there are the genome to express those things is inside of that space. It's kind of an interesting thing about the mitochondria, right? So I should just, as we're thinking about this, and we got this simple picture here. Remember that, just taking you back to cellular respiration, you kind of want to have a good picture of that. So cellular respiration, remember we start with the sugar, and then glycolysis is the breakdown of that sugar into 2 3-carbon molecules. That happens in the cytosol of the cell, so out in here. That's where that's happening. Then we have to transport the pyruvate, or the 3-carbon molecule, into this. So it has to go through that membrane. And then once it's transported in, it goes into the matrix, or the most inner compartment, and that's where the citric acid cycle happens. Okay, so all of those enzymes have to be in there. And the pyruvate itself has to get in there. Okay, and then the ATP gets produced, right? We use this inner membrane to build chemiosmotic pressure, because we pump protons into this space so that they can diffuse back down into the mitochondrial matrix, and we can produce ATP, and then that ATP has to get out. Okay, so we're going to talk about the proteins and getting them in there, and then we're going to talk about how those other things get moved in. So here it is. This is the more detailed picture of what's going on. It's worth repeating. So here's our outer membrane, and there's the intermembrane space. Here is our here's our inner membrane, and here are those electron transport chains for oxidative phosphorylation. And we can see in the picture, we're kind of going upside down here, so we can see in the picture that we have these proton pumps to build the proton gradient, and then these have to flow from high concentration to low concentration, and they choose to go from this concentration, from this space to this space, rather than this space to that space. That's another very important thing, right? So they go into the mitochondrial matrix. And by doing that, they crank this ATP synthase, and they produce the bulk of our ATP that we use for all of the work we do in our cell. Okay? Oh, we didn't mention the electron carriers, these NADH and FADH2. A lot of it comes from the citric acid cycle from inside of the mitochondrial matrix. A few molecules come from glycolysis, which is outside, and that they have to find their way into. So lots of stuff going on. So, I should point out, so the inner membrane is impermeable to to ions. The membrane composition is such that it is pretty much impermeable to any ion. And the only way in, so that means that the only way in for that H plus proton is through the ATP synthase. And that's a really important feature of this process. It has to be the only way in for that. An interesting thing about this is that about the outer membrane is that this is actually kind of porous. So there are lots of channels within the outer membrane that allow the cytoplasmic contents to flow into the inner membrane space. And actually, the cytoplasm and its protein and molecular signature is very similar to what is in this inner membrane space. So the cytoplasm and the inner membrane space have very similar content to it. Okay. But I should say that while there are lots of channels to help move things into this space, there is a membrane composition that prevents the H plus protons from going the opposite direction. So there's a very specialized membrane lipid that, kind of repels or directs it the other direction. Okay, maybe repel isn't the best word, but it prevents it from coming out out this way. All right. The other thing we want to remember or know about the mitochondria is there are many of them, and they actually form this interconnected network within the cell. This is actually a red stained image of the mitochondria. It's really kind of fuzzy, I know, because it's like the mitochondria are super tiny, and a lot of times, like, we'll do a staining like this, and they'll be like, oh, this doesn't look right, because you're looking for these, like, discrete little puncta that look like bean-shaped things. But the truth is, the mitochondria are fusing with each other and dividing, and they're kind of like, they're malleable, like, maybe if that's a good word, but they increase in number, they decrease, all kinds of stuff. The other thing is, they're kind of sitting on our cytoskeleton. They're actually like connected to our cytoskeleton in a way, so that they can walk along and be kind of tethered within the cytoplasm. They don't just float in the cytoplasm. Okay. So you're seeing them like it looks like fibrous because they're kind of like along the fibers of the cytoskeleton. So that's an interesting thing. They divide. I actually had this question in Bio 1 yesterday. So, because we were talking about cell division, so they were asking about the mitochondria. How does that work? Do they divide too? Or how are they separated in daughter cells, and how do you get more of them? So it's an interesting thing that they do it themselves. So they go through fission. Prokaryotes go through binary fission. That's how they divide. And so they do a very similar thing. So they replicate their genome, and then they can divide. So that's another piece of evidence that leans on that theory of endosymbiosis, that they were once a bacteria by themselves. All right, so they have their own DNA, and what does that look like? It looks like a prokaryote's DNA, so it's a single circular molecule is the whole genome for it. And there's some of the genes are mapped out for right here, so you can see them. But they are, it only codes for a couple of proteins, and the proteins that it codes for are the ones involved in oxidative phosphorylation. So those transport, those proton pumps, the ATP synthase, those things it's expressing. It also codes for its own ribosomal RNAs because the mitochondria have their own ribosome as well, right? So they really are almost like a living cell within the cell. But I will say that why is it an symbiotic relationship? It's because there are around 37 genes on this genome, but there are only 13 proteins that are produced by the mitochondria's own genome, and about 1,500 proteins come from the cell's nucleus. So the nuclear genome of the cell. So while it's got its own genome, it doesn't do everything by itself. All the proteins that build the structure of the ribosome, of the mitochondria, it's not coming from this genome. They're coming from the cell too. So different species, different eukaryotic cells have different sizes of mitochondrial genomes. From species to species, the genome differs. Another interesting fact about the mitochondrial genome is this is inherited by your mother only. I've brought this up before, right? Because the oocyte has the mitochondria from the mom, the maternal mitochondria. The sperm doesn't bring a single mitochondria with it when it fuses with the oocyte. All right, one more thing about the mitochondrial genome that's really unique too is that we have the way that the proteins are translated is different. So the codons are a little bit different in for the mitochondria. So where in the human genome we have the stop codons UGU or UGG or AG. Sorry, I'm saying it's AGA, AGG are stop codons in the human. I am saying this completely wrong. Sorry, guys. UGA is a stop codon for our nucleus, for our universal code. But in the mitochondria, UGA codes for tryptophan. AGA and AGG are the stop codons within the mitochondria where they give us arginine in the universal code. Sorry. We're still waking up. So yeah, that's another interesting thing, right? So it's like a different code for the proteins. So it's really a kind of a weird thing. There's one more thing I wanted to say. Okay, so yeah, just wanted to highlight that one more time. So we get like 13 proteins from the mitochondria, but we get over 1,000 from the cell. So now this mechanism that we're going to talk about, I want to make it clear that when we talk about transporting these proteins, these are the ones that are coming from the cell's nucleus, right? So they get expressed in the mRNA gets built, in the nucleus of the cell. It gets transported through the nuclear pore complex, and then it has to be translated on free ribosomes in the cytosol, and then it comes to the mitochondria. So now this is one of our first pictures of the mechanism for bringing the polypeptide that belongs in the mitochondria into the mitochondria. So, most of our proteins are synthesized on those free ribosomes, and then they come to the outer membrane of the mitochondria. They have to come through we have a double membrane structure, so we have to go through sort of a complex import into the mitochondria. So in our first picture, we have, our molecular players, we have the Tom complex, which is the I'm gonna say it wrong, the translocase of the outer membrane, so that's where Tom comes from. And then the inner membrane, you can guess it is translocase of the inner membrane, so Tom and Tim. Okay, and if you're thinking about, you know, follow the protein, it goes Tom first and Tim next. Okay, when we're importing. So here is our protein. It's coated with those heat shock proteins, so HSP70 specifically, and it's binding all of those hydrophobic regions so that this thing stays linear and it doesn't fold, right? Because we've got to get it through a membrane. It has a pre-sequence. We've seen these before, right? Those nuclear localization sequences, the sequence that helps to direct things in through the endoplasmic reticulum. We've got the same thing here in the mitochondria. So we've got a bunch of positive charges on this thing, and it is attracted to this like I don't want to say it's a C-terminal tail, but this structure that's positioned in the intermembrane space, on the intermembrane space side of the Tom complex. So this is essentially a channel, and it is an ATP, it's work, so some ATP is expended to move this thing into the channel, and this is attracted to that part to pull it through. All right, once it's in if it is a matrix, if this is a matrix residing protein, it has to leave the Tom complex and then come in through the Tim complex. So if it is a matrix residing protein, so TIM23 TIM23 is really interesting because it has the ability to pull a matrix protein all the way through. It also has that ability, remember our translocon, we could move it out of the way and the transmembrane proteins could slide right through into the membrane. Okay, so it can do both things. So we have our matrix protein coming through, and on the inside in the matrix we have this MPP, which is let me say it right, a matrix processing peptidase. Matrix processing peptidase. So what this is doing is it's cleaving that pre-sequence and it's pulling, it's cleaving the pre-sequence and it's pulling the matrix polypeptide into the matrix. Notice the position of some more HSP70 chaperones right there. So they go ahead, because they're released out here and this thing is getting pulled through, and then as it comes into this liquid space, then we have return of the HSP70 so that it can go and get properly folded once it's inside. All right, this one over here came through and it had a hydrophobic alpha helix, and that hydrophobic alpha helix drove it, instead of coming through, it drove it into the membrane. Okay, so TIM23 reorganizes and lets the transmembrane protein come through. This is how the single pass transmembrane proteins for the inner membrane work. It's going to be different when we have multi-pass proteins, when there's several transmembrane domains. There's a different way it's done. All right, one more thing to point out in the picture is notice that we've got these positive charges and these negative charges in here. Why is that? So the inner membrane of the mitochondria pretty much always carries this polarity in its membrane. And the reason is because remember how the oxidative phosphorylation mechanism works. We're constantly pumping H+ protons into this space. Okay, those bind up all of the negatively charged phospholipids, or they kind of are attracted to them, and they put this positive charge on that side of that membrane. In relation to that, this is negatively charged. So notice we've got this positively charged pre-sequence. It's actually attracted to the matrix because of that positive charge. Okay. It's not really excited to be in this space because of all the positive charges, right? So it's kind of a driving force to push it all the way into the matrix. Does that make sense? That's one of those things that when I took this class many years ago, I totally just missed. I never really understood that. So let me know if you have any questions about that. Okay, but positive and negative charges drive a lot of things. a lot of movement. All right, so that's how a matrix protein works and a single pass inner membrane protein works in the mitochondria. All right, in these pictures, we have the protein, a protein that's destined for the outer membrane. We'll stick with that, this side first, okay? So we have our, here it is in the cytosol, it's coated with HSP70, the chaperone. If it belongs in the inner membrane space, it comes through the Tom complex, so it looks the same as our last picture. And then there are these key chaperones in the Tom complex that will, so these fall off and these will bind. This is TIM9 and TIM10. So TIM9 and TIM10. This is where we get a little confusing in the mitochondria, because it was TIM23. So we start to have these numbers now that you want to kind of keep in mind. But TIM23 was in the inner membrane, was a translocator. The TIM9 and TIM10 are these little binding proteins for intermembrane space proteins. Okay, so intermembrane. Or sorry, yeah, the intermembrane space. Not the N, no, that's the T. All right, so the TIM9 and TIM10 help us to fold these proteins that belong in this space so they can be properly folded. Other proteins will come into this space and or come into the Tom complex and, other proteins will bypass the Tom complex if they belong within the membrane of the outer membrane. So the translocator or the channel protein that helps to put a single pass transmembrane protein in the outer membrane is MIM and MIM1, and I did not write down the full name for that, but MIM1, oh yeah, is the transporter for that. Okay, so MIM1 works just like we've seen with the other ones. It's a channel and it reorganizes so that the protein can move laterally into the membrane. But this one specifically lives in the outer membrane. All right, this one over here, what's going on with this one? We have the SAM complex. This is the sorting and assembly machinery complex. And the SAM complex is one that helps when we have a very specific structure in the outer membrane. This isn't really common. We haven't seen this a lot. There is typically we see alpha helices, but beta barrels can fit inside of the membrane as well and be transmembrane. So in this example, the SAM complex is going to help feed this into the membrane. So notice that we have this protein has come in through the Tom complex, or the peptide has come in through the Tom complex. It has bound the TIM9 and TIM10, and then it's moved into the SAM complex and put into with the help of the SAM complex, it's folded into the beta barrel and into the outer membrane. Sometimes I have to talk through these things to realize I'm saying something wrong. So one thing that I have said wrong is I mentioned that the TIM9 and TIM10 were helping fold proteins. It's a different set of chaperones, so I apologize for that. So this protein here is with chaperone proteins, probably another heat shock protein, a different number. Okay, did you catch that? Okay, so it's TIM9 and TIM10 that are helping to move these into the membrane from they're coming from the inner membrane space rather than the cytosol for SAM. Is that crazy? There's a lot of mechanisms. So next week we're going to spend time drawing these with our groups together, and we'll have some drawings to look through. But that's one of the best ways to really lock in what's going on and what the players are. I'm not going to ask you to draw these on the test, but I might be asking, if the TIM complex is blocked, then what's going to happen to a protein that belongs in this space or something like that. Okay. All right. Let's go on to this one. Follow the diagram here. This is a protein that is So we're looking now at proteins that are going to go into the inner membrane, and they are a transmembrane protein. And they can come from a couple of different places. So they can come from the cytosol. They can also come from the genome within the mitochondria as well. So following this one, we have movement of this polypeptide through TOM, the outer membrane complex. And then we have TIM9 and TIM10 again. There's going to be some sequence that's going to direct it it's not going to go to SAM. TIM9 and TIM10 will work with in this scenario, TIM22. So TIM22 is going to receive the peptide, and then it will move it laterally into the inner membrane space. And this is specifically for when we have a tetra spanning, a multi-pass alpha helix protein in the inner membrane. Okay, this one is coming out of our mitochondrial ribosome, so it's being expressed by that genome. We have HSP70 also in the matrix of our mitochondria, and this is going to be the chaperone that takes it to OXO1 translocase. So OXO1 translocase is our transmembrane transporter for the proteins that are coming from the mitochondrial genome. And this helps us to get that single pass membrane protein in there. Does that make sense? So now we've looked at a couple of things. We've looked at, let's just run through. We've looked at the movement of a cytosolic protein that came from the nuclear genome into the matrix and into the inner membrane. Now we've got cytosolic proteins that are coming into the intermembrane space or into the outer membrane if they're single pass. And we have cytosolic proteins that come into the inner membrane space, and then they get directed back into the outer membrane for a really complicated secondary structure that's built in the membrane. All right, and then over here we've got our cytosolic protein that is coming from the nuclear genome and coming through the outer membrane into the inner membrane and getting positioned within the inner membrane. And then finally we have our mitochondrial derived polypeptide, so coming from the mitochondrial genome into the inner membrane space. So I'm going to tease out those mechanisms for our exercise next week. So different groups on different things. All right, so that's how we're moving proteins. We also have to have lipids that are brought. So notice there isn't an endoplasmic reticulum really in our mitochondria. And while we can, build lipids, like so prokaryotes actually they divide, they can build lipids too. For the mitochondria, the lipids are actually coming from some of the lipids are going to come from the smooth endoplasmic reticulum. Okay, so here is our endoplasmic reticulum lumen. This is that smooth ER. We build the phospholipid in the endoplasmic reticulum, and then there is a transport protein that grabs it from the endoplasmic reticulum and carries it to the mitochondrial membrane. So here we have, it's called, do we name it? We don't need to name it. The phospholipid transfer proteins. Oh, it's right there. Phospholipid transfer proteins grab the phospholipids from the smooth ER and transport them and stick them into the outer membrane of the mitochondria. That's how they get there. Now the outer membrane of the mitochondria, remember that it's important, it has a lot of pores to allow cytosolic contents to freely pass in and out of it. But it does have specialized lipids that prevent that H+ proton from just going through it, okay? So we have, the specialized lipid is cardiolipin. So notice what cardiolipin looks like. We've got our glycerol, 2 of them, and here are our 2 fatty acid chains, and then we have this structure that kind of bridges them together. So it's like a little bridge in between. So it's a barrier to the H+ proton. It's an additional barrier. We're not going to go into a ton of detail about that, but does that make sense? So it's a special, you want to remember there's a specialized lipid called cardiolipin in the outer membrane that restricts that proton flow across the membrane. All right, this is our last slide for mitochondria. So we started off remembering what happens in the mitochondria and how we move all this, like we have, you know, we have to bring in the pyruvate and the electron carriers and all of that stuff, and then we have to move the ATP out, right? So, and then where is the citric acid cycle? It's happening in the matrix, the innermost compartment of the mitochondria. So we have a lot of small molecule transporters in the cell. These are separate, or in the mitochondria, these are separate from those transporters that we were looking at that were, you know, the protein transporters. So let's see if I can remember which ones I'm talking about here. Okay, so we have, right here, we have an ADP-ATP transporter. So this is a really interesting one because the way it works is it's one ADP in, one ATP out. So that's the regulation. It brings one ADP in for and then there's one ATP that goes out. Okay. The electrochemical gradient is generated by our proton pumps. Remember in the proton transport chain, so this is in the diagram is showing us the high concentration of H plus and the low concentration of H plus in the matrix. So because we have all these pluses here, we've got that polarity to the membrane. We want to remember that polarity because that drives a lot of that small molecule movement as well. All right, so we have inorganic phosphate and an OH, well, inorganic phosphate transporter. So we bring them in and then what goes out is an H plus proton, I think is what goes out for that one. So it's another transporter. Why is that? Because remember, like somewhere way out here in the intermembrane space, we have to join some ADP that is being brought in with that, right? So that's an interesting thing too. Those two ingredients come in through their own transporter. So they both got to be working for us to make ATP. All right, and then pyruvate has its own transporter as well. So we have pyruvate that comes in through a specific transporter. So those are the things we needed, right? The pyruvate, ADP. What isn't pictured here is the NADH from glycolysis and that, but that also has its way in. It has a small molecule transporter. We don't really need an FADH2 transporter. Why? Because it's getting generated in here, in the matrix, right? We get NADH from glycolysis. We get NADH and FADH2 from the citric acid cycle inside. All right, so that's where I wanted to end. If you guys want to take the 10 minutes to talk with your group about where you want to meet on Friday, do that. But Friday, again, is all day.